Again, our balanced sodium and potassium, how much is inside, how much is outside, is setting up this electrical difference. Once the nerve has fired, it relatively slowly returns to the resting state. Once it's fired, it can't immediately send another signal. It has to wait until it's able to reset again. Again, you've got those gates that open or close. You have to get that all reset, and then it's ready, but it won't be as ready right away as if it's got more time in between. So it takes a stronger signal to get the neuron to fire again faster than the signal that first made it fire. Again, the sending a signal on for one neuron is all or nothing, yes, no. Did I get enough of a signal coming in to say pass on a signal to the next part? Then it will. If it doesn't get the input, then it won't. Neurons cannot say, I'm picking up a weak signal here. Hey, we're getting a little bit. No, it's going to be either yes or no. Does it fire or not? Okay, but you've probably noticed you feel some things more than others. So obviously you're getting more signal in. That relates to how many neurons are picking up a signal and how quickly. Do they keep on getting that signal and responding as soon as they've recovered or not? So this nerve signal sending is traveling as a wave of the polarity difference. Again, remember our cell is more negative on the inside, more positive on the outside with more sodium. But when gates in one spot open, we get the sodium in, that changes potential next to it, and so it moves along. Again, the nerves that don't have that extra insulation layer around them, it just travels all the way along. But if it has that extra insulation, it can basically jump from one gap to the next in the insulation and travel faster. Saltatory, that's just a fancy word for jumping. The charge difference hops along from one to the next and travels faster. Okay, then we get to the end of that neuron, have to get the signal to the next. Sometimes you actually have direct connection between the neurons and the electrical signal can just go right from one to the next. But more often, there's a gap in between them where chemicals travel in between. And so this provides another way of regulating what's going on in the process, and then the next cell receives those chemicals and uses that to determine its response. Examples, there are a number of different types of chemicals used as neurotransmitters. Even some simple ones like nitric oxide, carbon monoxide. Those are both things that you don't want large quantities of, but in tiny amounts in the cell, they're actually used regulating signaling. So basically, at the end of our nerve, we have vesicles inside the cell holding neurotransmitter, available if it's needed. If that electrical signal gets to the end there, that triggers moving those vesicles so that they fuse with the outside the cell, exocytosis. We release the neurotransmitter, moves along, reaches receptors on the next cell. Of course, this is a system that can be interfered with. A number of types of venom used by predatory animals messes with the reception of neurotransmitters. So the thing that gets the venom into it doesn't have the nerves responding properly and will be helpless and easy to eat. Okay, we've released the neurotransmitters. They get to the other cell. Now that could be a signal for that cell to do something or to not do something. They could be excitatory or inhibitory, making it more likely to send a signal on or do something, or less likely. Depends on that cell and its receptors. Now, if a cell is getting some of both, some saying do something, some don't do something, well, how does it add up? Is it getting about equal from both? Or does the do something signal get bigger enough to actually cause something to happen? It'll just be how it adds up. This can add up in two ways. It may be if you have more and more signals coming in and decide eventually you need to do something. Or it could be lots of them, say multiple neurons all signaling at the same time, giving a big impact. Either way, like our previous neuron, you reach the right threshold and boom, there goes our electrical signal along it. Assuming we're looking at a neuron rather than the muscle or other possible things. Okay, but now that neuron needs to get ready for the next directions. So we have to clear out the neurotransmitter from the space between the cells. Generally, there's some way of bringing it back to the starting cells so that it will have that ready for the next signal. Neurotransmitters often are affected by various drugs. These include both legitimate drug use medically for particular conditions, but also a lot of the ones that are abused recreationally do things to the neurotransmission, which on the one hand may give you some sort of exciting feeling, but it's messing with the connections in your brain, which is not good. Lots of neurons interacting for the central nervous system. In something like our cells, we have millions of neurons, very complex. And so each particular neuron has its proper place. Connecting in, we can have several neurons connecting to one neuron that puts all that information together. We can also have neurons that connect out to multiple ones, sending out the same starting signal to multiple different places. Okay, all this information is coming in. Central nervous system has to process it, decide what to do, and then send a response signal back out. In addition to data from outside, your brain needs to also keep track of, okay, you're still breathing, good, heart beating, maintaining all the standard functions of the body. So we have both the internal monitoring and responding to all the signals from outside. Some organisms are much more simple. For example, cnidarians, typically the nervous system is just kind of net-like. And so one neuron gets a signal and it sends it around to other neurons. But just kind of all around, there's no central processing. Just kind of, hey, I've been poked, okay, squinch up, move away from here. Those types of things. Bilaterian animals, ones that have front and back, left and right, characteristically have a somewhat more centralized nervous system with something roughly equivalent to a brain. Now, often it's pretty simple, like the cerebral ganglia here in this flatworm. Rather small, simple components, but it's a little bit of concentration of nerves. It does function in decisions there. And then you have main nerve cords with further branches extending in the rest of the body. This happens to be a flatworm planarian. A little more complex, here's an arthropod, a grasshopper. It's got a little brain, a ventral nerve cord. Our main nerve cord is dorsal. And these ganglia, these are little bumps, concentration of nerves. Often there's some signal processing in the ganglia as well. Among invertebrates, the most impressive brain and intelligence abilities are found in cephalopod mollusks, octopus, squid, things like that. They also have well-developed sense organs, eyes functioning quite well, similar to ours. The brain forms a ring around the esophagus, the top part of the digestive system. This does have problems if they try to swallow something that's too big and it pokes out. Brain's kind of vulnerable to that. But it has complexity, kind of like ours, folds, different lobes, and doing different things, coordinating quite a wide range of actions. Octopi have quite impressive ability to learn, figure out relatively difficult tasks.